Okay, guys, thank you so much for coming. So today we're going to review for midterm 2. We're going to do lecture 6 through 10, okay? And then we'll kind of go through a little bit more detail for the muscle contraction lecture, okay? As usual, the exam format is going to be the same as last time, all right? We're going to have our usual section 1. And this is the multiple choice, right? Okay, and it's going to be 30 times 1, which is equal to 30 points. And then section 2 is going to be the short answer questions. Okay, and you answer 5 of 6 times 2, which is 10 points. All right, in section 1 here, when I pick the multiple choice questions, I pick a question and then I pick one for the quiz and then one for the midterm, so that they will be related. So when you do your quiz, okay, you can print the answers, and the quiz will also give you like all the information of how to work out the answers. And so if you study around all those 30 multiple choice questions, chances are that it's going to be the same kind of questions, but it will be a different question, but it will be in the same subject area. So
it will be the same question, just
yeah, because when I pick them, I pick them one after the next. So I pick a quiz question, then I pick a midterm. So they are the questions next to each other are mostly around the same subject area. So if you do your quiz and you print it out and you look at the solutions and how to work it out, and you look back in your notes or your textbook, that way you study around the area that I'm going to ask for. So you stand a good chance of getting 30 out of 30 points for your midterm. You see what I'm saying? So that's why I do that. When you do the quiz, I'm going to post it tonight. Just know that if you study around that area, it will help you, okay, with your multiple choice. So you should really aim to get all the multiple choice questions right for the midterm. That's about half of your exam, 30 out of 60 points. You see what I'm saying? And then the other ones, some of you may have noticed, but there are some multiple choice questions that have pretty much answers for some of the long answer questions. You know what I'm saying? So almost always do the multiple choice first, then you go to the short and long answer questions. Okay, so I do that deliberately, and sometimes it gives you like 3 points in terms of answers. Okay, times 5, which is 20, so that's 60 points total. All right. So make sure you definitely do those questions instead of the multiple choice. All right. So the first one was bone physiology 1, right? I mostly talked about the structure of bones and the components of it. We should all know the components, right? The tendons, the ligaments, okay, and the bones and the cartilage. All right, so those are the components. And of course, the bone is living tissue, so you've got nerves, okay, and also have vascular tissue there to bring in the nutrients, oxygen, and take out carbon dioxide, okay? So just that's the basic things about. Of course, you should know the functions. Remember, we talked about how it supports, how it, So these first lectures, I'm kind of going to go a little bit quickly to reserve a bit of time so I can do that muscle contraction in a little bit more detail. Because the muscle contraction really is a little complicated, and you have to know all those proteins that are associated with that. Okay, so I'll kind of try to go through. So the support, we know about that, the muscle, the skeleton, and then the body, the protection, like the brain and so forth, the vertebral. So you explain that storage, calcium, right? You explain it. In the exam, you kind of flesh it out a little bit, and then movement, of course, muscles attached to the skeleton, and the skeleton acts as levers, and then of course blood cell production, all the blood cells originate in red bone marrow, right? Okay, so you talk about that. If I ever ask, sometimes I say list the 5 functions of the skeletal system, and then you talk about that, and you explain that. Most of the time, you just have to think about what the skeleton is doing for you, right? Okay, so we know about cartilage, which is the model for bones that are endochondral ossification, So it's got chondroblasts. All right. What do chondroblasts do?
Make new.
Yeah. New matrix, right? Okay, so you gotta know the cells. That's one of the things about the skeletal system. Know the cells and know what the cells are doing. Blasts form something. Site is a mature cell. After the chondroblasts have produced the matrix, they become mature bone cell cartilage cells called chondrocytes. Okay. So these are mature cells. Okay, so you gotta get, and then of course the cartilage matrix has got the collagen fibers, okay, made by the chondroblasts and fibroblasts, and then the proteoglycans. What do proteoglycans do? They absorb water, and therefore the cartilage can be compressed, and it goes back to its original shape. Okay, these absorb water. Okay, and the collagen of fibers, of course, give it the strength. Okay, so you gotta know the basic components, the basic cells, and then of course the matrix, because pretty much connective tissue is all matrix, very little cells. Okay, so you gotta know about the matrix, and then of course the perichondrium is the outside membrane. All right, the outer layer is what fibroblasts. Okay, and then the inner layer is chondroblasts. Okay, and the chondroblast makes the matrix. The inner layer is one that produces the matrix, and then the outer layer is one that produces the connective tissue covering on the outside. I mean, this is the connective tissue that in the future bone will house the blood vessels. You know what I'm saying? Because the cartilage is the model for future bones in endochondral ossification. Okay, so know about the membrane covering cartilage is a perichondrium, chondrium for cartilage right there. All right, and then of course the growth of cartilage. Right? What are the two types of growth that we talked about? First one starts with an A. Oppositional. Oppositional is what kind of growth? You need to know that. On the and it grows on the surface of the bone. Okay, you add a new matrix on the surface of the bone. So usually this is one that increases width. Okay, it increases the size of the epiphyses, which are the ends of the bones. Right? And then what else? Interstitial growth. Interstitial, by the meaning of the English word, between, within tissues, right? So this is the kind of growth that occurs where at? Internally, where at? There's a certain part in the epiphysis called the epiphyseal plate. Remember, the epiphyseal plate remains as cartilage, so you form new matrix from that. Most of the bone, in the young bone, that is calcified and ossified, right? But it's the certain part in the epiphysis called the epiphyseal plate where the interstitial growth occurs, okay? So you gotta know interstitial growth is the definition. You gotta know oppositional growth. In the 2-point question, sometimes I'll sprinkle that and say describe oppositional growth. Describe interstitial growth. Get 2 points, right? All right, so there you go. So we know bone is got, 35%, the matrix. Organic, and then this one is, okay, it's mostly minerals, of course. So gotta know the bone, the composition of the bone matrix in terms of the organic part and the inorganic part. The inorganic is the minerals. The organic part is things like the proteoglycans and the collagen fibers, right? And know what the significance of the organic matter is. Know what the significance of the inorganic matter is, right? Remember when I showed you that bone that was brittle because it had too much inorganic and not enough organic? If it's got too much organic, it's just like limp actually. It's not going to do the support. Okay, so be able to, I may ask with the components of the, what are the percentages of the 2 major components of, bone matrix, and describe their significance or their importance. And then you talk about that brittle bone or that very flexible bone. So you get that, okay? And then of course the bone cells, Now we're going to the bone, and then osteo means bone. And then the same kind of meaning, the blasts will form the new matrix. After they form the new matrix, they become mature cells. And then now we add new cells, because bone can be remodeled. Okay? The bone can be remodeled. So we have osteoclast. What is the function of osteoclast?
It breaks down.
It breaks down. So this is my shortcut for break. Okay, break down. Okay? So then I know, again, the cells, and I know what their function is. Know the matrix. What are the components of the matrix? Okay? All right, so, and then osteogenesis, of course, formation of new bone, or ossification, sometimes it's called. All right? And it's got those 3 steps where you have the osteoblast on the outside. You know how their processes meet and they form kind of tentacles like that, and then the spaces in between gets filled with matrix. And then you form vesicles filled in with the proteoglycans and collagen fibers, and then secreted outside of the cell. All right? And then what happens? Because the cells are like that, right? And then they meet like that. Okay, so this space is like that. You get those first vesicles that are formed. If I ever ask you to describe osteogenesis, then you talk about the first vesicles that come from these osteoblasts, all right, via exocytosis carrying the matrix. And the matrix is collagen fibers and proteoglycans. And then it comes out here, and then it goes into those spaces, all right? And then the second vesicles in osteogenesis, what are they carrying? What is the component of matrix? We said we got the organic part has been taken care of. We've got the collagen and the proteoglycans. What's the next part? Minerals. Okay, so it's going to contain calcium, hydroxyapatite, and phosphorus, right? Okay, so those 2 matrices, you know, you got to talk about that, the 2 matrices. The first one carrying the organic material, the collagen and the proteoglycans, and the second vesicles carrying the minerals. You first of all put your outline, then you dump the mineral on it. The mineral is like the concrete, okay? So that comes second. So if you talk about osteogenesis, you have to mention those 2 matrices. That's plural for matrix, okay? You get the first vesicle, 2 vesicles. Okay, 2 vesicles. The first one carrying organic matter, the second one carrying mineral. Okay? All right. This is on the surface, okay? It's occurring on the surface. So, oppositional. Okay, and then, of course, you first of all make woven bone. You know what woven bone is? Fibers all over. Okay? The collagen fibers are oriented in different directions. All right? And then, lamellar bone, you then break down that woven bone, and then you layer it, right? And you form those 3 layers. You get the circumferential lamellae on the outside, the concentric lamellae that go around, and the interstitial lamellae that fills in the spaces between those osteoles, right? Okay, so you're going to be able to describe woven bone, lamellar bone, right? Okay, and then of course the 3 cells, and then osteogenesis, that was 3 steps. And then the way bone is classified is the cancellous bone. It's got trabeculae or beams in it, and it's got more spaces than it has matrix, right? Okay, and those trabeculae beams are not just haphazardly arranged; they are, you know, oriented along the lines of stress, so that makes it strong. Okay, trabeculae beams of the matrix, and they still go by appositional. They go by appositional core. And then, of course, the compact bone, where you have the osteons. So you have those lamellae that we just talked about, circumferential lamellae on the outside. And then you get the interstitial lamellae, of course, that fill the spaces between the circles, because the circles don't meet like squares do. And then, of course, the concentric that goes round. Okay, concentric is going around the central canal. All right, and then, of course, the osteon. All right, so the osteon, we will later on talk about the structure of the osteon, all right, with the central canal in the middle carrying the blood vessel, and then the perforating canals that come from the outside, Remember, the fibrous membrane on the outside of the bone is carrying the blood vessels, right? So the osteon is like that. It's like a cylinder. And then in the middle will be the blood vessels, all right? And then you've got another osteon here. Okay, and then on the outside you have the membrane carrying the blood vessels, and then you have the Volkmann's canals that cross to go to the central canal, right? To provide the blood. And then in the concentric lamellae, you have those canaliculi, which are small little cracks, you know, that will take like that, right? So these are concentric lamellae, and sometimes you have a cell here, a cell there, a cell there. So the cracks in there are the canaliculi that will distribute the nutrients throughout the osteon, right? Okay? So you should know the components of the osteon in terms of the central canal with the blood vessel, the perforating canals, kind of like the microscopic structure of the osteon of the bone, because the osteon is a unit of compact bone. Okay? And then you have those cylinders all together like that, filled with interstitial lining. So to know those canals, and their importance, of course, is to distribute nutrients and gas, okay, oxygen, and take out carbon dioxide. Okay? So that's the compact bone. So we get the central canal, the Volkmann's canal or perforating canal coming from the side of the bone, carrying blood to the central canal. And then the central canal spreads the blood throughout the osteon via the canaliculi, which are smaller, you know, channels. All right. So go through your notes. Of course, the basic structure of long bones is that it's got the ends, right? I like to draw my dog bone. Okay? And then in a young bone, in the epiphysis, you have the epiphyseal plate over there, where the interstitial growth is occurring, right? Remember? And then the length of it is the diaphysis. Diaphysis, and up here, the epiphysis, okay? The ends of the bone. All right? And in the middle, what do you have? What's it called? What's that middle cavity in the middle? Starts with an M. What is it called? Do? Guys, I've given you the whole word now. Medullary cavity, right?
Yeah.
Okay, guys, medullary cavity. Because you should have kind of been reading.
I was going to say that, but I was thinking that was in the kidney.
It's in the kidney too. So that's the thing, and it's all over the place. The kidneys, the medulla, the adrenal glands have the medulla. All of them have the medulla. It's just a layer. Okay, so the medullary cavity is in the middle, and it's got spongy bone, and it's got the bone marrow inside of it. And what's the membrane that covers the medulla? We'll talk about it in a little bit. The membrane that covers here. What's it called? What's my other friend? Oh, there it is. Ah, okay. What's that? Endosteum. Mycelium is for muscles and stuff like that. Osteum is bones. Osteum and osteo, you know, is bone. Okay? And chondro, cartilage. Part of bone. Bone starts off as cartilage and then becomes osteum. So, endosteum. And then on the outside, on the outside is another membrane covering the bone, which is the periosteum. Okay. All right, guys. So go through your notes, guys, and kind of learn these things. Those are them, right? Those membranes. So this one is the outside one, and we said it's got the fibrous layer on the outside carrying what? The blood vessels. Because the blood vessels, they go along the connective tissue. That's the same function of connective tissue in muscle. Did you notice that? Connective tissue surrounds all these muscle bundles, and then the blood vessels go along the connective tissue. So the same thing in the bones, same thing. That's the good thing about the body. You learn thing once, you apply it to a different situation, it's the same function, okay? Because it's the same structure. Okay, structure is always related to function. Okay, so the periosteum on the outside, carries the blood vessels, and then of course it's got the fibrous one on the outside with the blood vessels. So this is the blood vessels, and then the osteogenic layers on the inside. Remember the membrane has got 2 layers on the outside, and then the osteogenic layer of course makes the bone. So it's got the osteoblasts, and then it's got the osteochondral progenitor cells. Okay, that changes the osteoblasts and chondroblasts. Okay, then it can tend to both. It's like a stem cell. So it is a stem cell, and that can be done those 2. So know these membranes, know where they are, they know what the function of the membranes are. Okay. Yeah. So this one is a fine one that lines the medullary cavity on the inside. Endo means inside. Perimeter on the outside. Right? Okay, so you gotta know these membranes because I'm gonna ask about them and I'm gonna say, okay, what's the periosteum and what's the function of it? Alright? So the periosteum has got 2 functions, right? It carries the blood vessels on the fibrous layer, and then it's got the osteogenic layers on the inside with the cells that produce matrix, like osteoblasts and osteochondral progenitor cells. Bone matrix too. Okay? So now this is talking about, the 2 kinds of bones, the flat ones, intramembranous membrane, which is ossification centers, and then it ossifies like that. And then, of course, the endochondral one, which is most of the bones of the body, long bones. All right? Most of the bones in the skull are the intramembranous ossification because they are flat bones, right? But the rest of the bones in the body are endochondral ossification, okay? And this one goes within membranes. Okay, it's centers of ossification, right? You see, this would be like a flat. Let's say this is a flat sheet of membrane, maybe the parietal bone or something. So you have a center here, and then it starts to ossify. You know what I'm saying? And then you have a center here, and then it starts to ossify and become, mineralized, produces matrix and so forth. And then another center starts to ossify. And then before you know it, the whole thing is covered with bone, membrane. It's a sheet. All right? And like the Fontanos we talk about it, those are parts of the membrane that starts off with intermembranous bonds that are not ossified. Okay, and then that will happen, after the fetus is born. It's now a neonate. Okay, so that's that. And this one, endochondral ossification, it starts off as models of cartilage models set off endochondral ossification, right? And we get the perichondrium on the outside, with the fibrous layer on the outside, and then the layer on the inside with chondroblasts and so forth that make matrix. Okay. So remember that long process where you get the perichondrium being invaded by blood vessels, which comes with nutrients, and then it starts to change these osteochondral progenitor cells into chondroblasts, which start to make new matrix. And then after that, you form the bone collar. Remember those steps? Because sometimes I will talk about ask about the steps of endochondral ossification. All right? After you form the bone collar on the outside here, so you start off on the diaphysis, right? You form the bone collar, this is the 3rd step. And then after that, you start to form primary ossification centers. You start to ossify, add the mineral to it. Okay. Then you get interstitial growth happening here at the ends where you start to form secondary ossification centers on the ends of it. Okay? And then you're remaining with a epiphyseal plate that doesn't ossify because it's going to be the future growth of the bone. Okay? So go back to your notes and look at all the steps in the endochondral ossification. Okay? So it uses highlight models, and most of the bones in the body are like that. So, all right. And then of course bone growth, we know about oppositional growth. It increases the size, like the diameter of the bone, because it grows on the outside. It's layered on the outside, and then of course oppositional growth and interstitial growth increases length, because at the epiphysis you've got that interstitial growth which is increasing length, and then also oppositional growth which increases the size of the epiphysis, right? Because the bone goes symmetrically in size, because as it grows longer, the ends also become bigger. So that's that. And then, of course, the epiphyseal plate is where the bone is the zone, the growth point for bone, right? And remember those zones we talked about, there's like 5 of them, right? We get the resting cells close to the epiphyseal plate. Then we get the cells that are multiplying, proliferation, the second layer. Then we get the cells that are hypertrophy, growing in size, okay? And then we get, calcification. Once you grow the cells, you get the matrix, and then you put the minerals, okay? And then, so that's calcification, that's the 4th one. And the last one is the ossification. So once you put that, it now becomes bone. That calcified cartilage now becomes bone. It's got all the 65% of inorganic material in it, and it's now bone. So that's ossification. And then the bone rests. Okay? So know those 5 zones. Okay? All right, so good. So growth at the articular cartilage. So there's different growths at the articular cartilage because some of these bones, if they don't have an epiphyseal plate, the cartilage, the epiphyseal cartilage will form the the model for the new bone, and then that's how it grows in size at the you still have appositional growth on the outside, because the outside of the epiphysis is compact bone. Compact bone will only grow by appositional growth. You know what I'm saying? Because compact bone has got no spaces in it, it's all packed up, it's tight. So you can only add material on the top of it. You see, you put it on top to make it bigger. All right? And then, so that's how you get those grooves growing along the blood vessels. Okay, so this is a blood vessel, and then you get a groove growing like that. Okay, and this is on the surface, right? And then these grooves meet like that. They form a central canal in the middle, and then the concentric lamellae is laid down, and then you form an osteum. So on the surface, let's say this is the surface of the long bone, looking at it from the side, you lay down a whole bunch of these osteons on the surface like that. And then in the middle of it will be interstitial tissue, the interstitial lamellae to fill it up. Okay, remember that we talked last time? So that's the growth there in bone width. Okay, so and then factors affecting bone, okay, like genetic, your genes determine how big your bones grow and the shape and form. Okay, so that's the basic part. What is the function of vitamin D? What is it involved with bones for? What does vitamin D do? What did we say last time? Vitamin D? I will give you a hint that is involved with the inorganic part. Okay? What's the inorganic part of bone? What is it? Calcium, right? Calcium is the inorganic. All the minerals are the inorganic part. And all the proteins and things like that are the organic part, right? So, what does vitamin do? It aids in calcium absorption, okay? So, calcium absorption, and we all know that calcium is the main component of the bone, right? Okay, you see? Okay. 10. Okay, and then 6, right? So 6 phosphorus, 10 calcium. That's the inorganic component. This is hydroxyapatite, okay? Hydroxy coming from this is the hydroxyl ion, 2. So that's hydroxyapatite. You see how much calcium is in there? So you need vitamin D to help in the absorption of calcium in the small intestine. You definitely need it, okay? So hormones, what hormones did you say affect bone growth? Like? One of them is involved in the females with menopause. Yeah, estrogen. So all the androgens, estrogen, testosterone is involved in bone growth. Okay, it causes bone to grow, and it also inhibits PTH, which we'll talk about when we talk about the homeostasis of bone. PTH is a hormone that breaks down bone, which is parathyroid hormone. Okay, so hormones like testosterone and estrogen will inhibit PTH, so that you don't break down bone. You build more than you break down. Okay. So if I ask about factors affecting growth, and I say, okay, explain 2 factors that affect bone growth. Then you talk about the inside genetic material. Then you talk about, well, yeah, you talk about vitamin D. Okay. PTH actually breaks it down, right? Okay. Then you could mention PTH insofar as the hormones are concerned. Because I might say, okay, for bonus, one more point, mention the 3rd factor that affects bone growth, right? Then you go to hormones, and you say, okay, hormones like testosterone and estrogen inhibit PTH, which breaks down bone, and encourage bone growth. And you get 3 points for 2. Right? I'm going to bring this question. I promise you. You see, I always bring it. And don't say I didn't tell you guys, okay? So I'm going to bring it. So you guys start this, okay? So I'm going to test you because you know, you kind of didn't seem to know some of these components. So I'm going to bring that question. So we want, okay, so remodeling, we talked about it. We said, you know, if bone, because bone is a solid thing, you gotta be able to break it down and reform it. Otherwise, if it gets messed up, you won't be able to do repair. Okay? So, you know, if I ever ask about the, you know, the significance or the importance of remodeling bone, so you talk about repair, you talk about, you know, being able to adapt. Okay? You know, and also you can break it down and finish calcium. So if you don't have enough calcium, you can break down the bone because it's, after all, the hydroxyapatite is 10 calcium, you know, ions in just one molecule of hydroxyapatite. So there's a lot of it. So calcium regulation, repair, and it's just to stress. It adapts. It makes the bone. It gives it the ability to adapt. Okay, and then of course it can change shape when you need to. So it makes it adaptable. What are the basic multicellular units? Basic multicellular units, what did we say they are assemblies of? They are temporary assemblies of what? What are they assemblies of? Well, basic multicellular units. The name says what they are. They are temporary aggregates of multicellular of cells, right? The name tells you what it is. So cells, what kind of cells? Cells that produce what? And do what? Cells that produce the matrix, right? Because you're remodeling bone. Okay, so what are the cells that produce the matrix? Osteoblasts, right? And what are cells that break down the matrix? Osteoclasts. So these are, you know, temporary assemblies, and we talked about how in about 10 years you have replaced your whole skeleton because you're constantly breaking down and laying down new bone. Okay? If I ask about basic multicellular units and say, you know, what do they do and what are they, then you say they are temporary assemblies of cells, such as osteoblasts and osteoclasts. Okay? You just have to talk about the cells. Remember I said at the beginning, know your cells. Know all those cells, what they do. Blast is making new matrix. Clast is breaking down. Site is a mature cell. After it has made the matrix, it becomes mature, and then it's an osteocyte. Okay? Or chondrocyte. So know about the basic multicellular units. What are they? Temporary compositions of cells comprising osteoclasts and osteoclasts. What is their function? To break down bone and lay down bone. They are for remodeling. Okay? So they are temporary assemblies of that. So know what they are. Okay? And then bone fractures, we talked about how they are classified according to the completeness of the break, or, you know, which part of the bone is broken and how, and they are important for bone repair and also, you know, treatment. So you know how to treat that, you know, bone. So that one just kind of, but you know what basic multicellular units are? What are they made up of? And what is their function? So bone repair, we got those 4 steps, right? We form, when you break bone, you're gonna damage blood vessels and you're gonna, you know, bleed, and then you form a clot, a big, huge clot called a hematoma, right? That's the first section. Okay, and then after that, when you have stopped the bleeding and you've got a hematoma there, and the blood has stopped bleeding, then you want to form granulation tissue now. You form callus. Okay, the granulation tissue will be those cells again that are going to make the new matrix repair, like fibroblasts, osteoblasts, and so forth. Making the new collagen, the new proteoglycans, and so forth, so that you can repair the bone. Okay, so osteoblasts, fibroblasts accumulate, and that accumulation of the cells is called granulation tissue. Okay, because this is just compiling the cells that are going to do the job of making the new matrix, and then of course the bone. Okay, callus ossification. So once you've got the matrix, remember bone is organic matter and inorganic matter. So always with bone, what you do, you make the organic matter first, which is the proteins, proteoglycans, collagen fibers, and so forth. Once you're done with that, what do? You dump the minerals on top of that to make it bone. So that's part where you add the calcium and phosphorus and so forth, the minerals, is the ossification. So then the callus ossifies, right? Because you've made your matrix. And then, of course, it always starts off as woven bone. Because when you make it, the collagen fibers will be all over the place, right? Because you're going to remodel, and in the end, you're going to break it down and then make it into those concentric lamellae, interstitial lamellae, and the concentric lamellae, right? So at this stage, it ossifies, and then of course the cancellous bone kind of forms first because it's got more spaces, and then the compact bone is formed late, because it's got less spaces. It's more compacted. Okay, and then the fourth step is then you remodel. Okay, then in terms of remodeling, you're breaking through, because the callus, it forms bone throughout the medullary cavity. So there will be no, opening on the bone at the site of injury. Okay, so then you break that down, right? Because you've got the osteoclast, right? So you break that down. So, okay, and remember you form external matrix that forms the external callus there to kind of protect the area that's being repaired, right? Then, at the end, you shave some of it off, okay? That's during the remodeling stage. The excess one on the exterior is also shaved off, but not all of it is shaved off. You still get a little bump on it, on the side of injury, and then you're done, okay? So if I ask you to tell me about the 4 steps in the bone repair, so you talk about that. So the hematoma formation, the callus formation, then after that you ossify, because you get out the organic material, ossification. Then after that you remodel, and you're done, right? And then when you remodel, you know what you're doing, right? You're removing the excess on the outside that we're using to protect the site of injury, and then you make your medullary cavity, so break it through. Because the callus just forms all over across like that. So the medullary cavity will be blocked from that side, and it's blocked. So you have to break through now. During the remodeling stage, you just go boom, boom, and then once again, okay? So know those.
How does that work though if it's flat bone?
Huh?
If you break flat bone, you're not going to create a new medullary cavity because it doesn't happen.
Well, what does flat bone have? It's like a sandwich. Remember, it's got compact bone on the outside, and it's got the cancellous bone in the middle. Remember, flat bone. So it's more or less the same, because then you have a little bit of spaces inside, because that's where it will be like the bone marrow, really, with all the blood vessels coming in through there and the bone marrow in it. That's why the flat bones of the hip, like the ilium, that's where you harvest red bone marrow if you are trying to donate. It's a flat bone, right? So it's still got that kind of cavity in the middle made of cancellous bones.
Oh.
You know what I'm saying? All right, so calcium homeostasis is very important because of the ability to remodel bone. You can have calcium homeostasis. So the bone is not just sitting there; it's constantly being broken down and laid down, broken down and laid down. Okay, which is resorption, breaking down, and deposited, which is laying down bone. Okay, so like we said at the beginning, what is the function of bone? What is the function of the skeletal system? One of them is calcium storage. Okay? If you were not able to remodel bone, then it will be no good. Doesn't matter how much calcium is in there. If you can, you know what I'm saying? If you can break it and get it out of there, you know, then might as well, you know, not even be there. Okay? So, the hormones that are important is especially parathyroid hormone, PTH, from the parathyroid gland. Okay? PTH stimulates osteoclasts, right? Because osteoclasts do what? Break down the bone to finish calcium, right? But it doesn't stop there, right? What else does PTH do? What else does PTH do? Anybody want to guess what PTH does? Apart from just breaking down the bone to finish calcium, what else does it do? It's going to increase reabsorption of calcium in the kidneys, right? Okay. What else does it do? It's got to do with vitamin D, right? What is vitamin D? Remember I told you about vitamin D. What does vitamin D do? It will, absorption of calcium. So it will increase absorption of calcium by forming active vitamin D. Okay. If it was only going to break down the bone, guess what would happen? At the end of the day, you end up with no bone at all. It's all brittle. So you don't want that. You want your bone to remain strong, so it will do a bunch of other things. So if I talk about in the calcium homeostasis, I say, okay, what are the 3 functions or what are the 2 functions of PTH? Then you would say, okay, PTH is parathyroid hormone, okay, and it will break down bone, you know, by activating osteoclasts, right? Then it will produce active vitamin D, so you absorb more calcium from the intestines, okay? Then it will also cause the kidneys to reabsorb more calcium so that you don't void it in the urine, right? So you reabsorb more calcium from the kidneys. So those 3 things, then you get 3 points, right? So that would be like a short answer question. You know? And then calcitonin, what does calcitonin do? It is involved in, we just talked about how that one is involved in breaking down bone. So this one is involved in making the bone, laying down bone, okay? So if you eat a meal with a bunch of calcium like milk, or if you drink milk, you're going to have a lot of calcium that's going to be in your blood. And then calcitonin is released so that that calcium is taken up by your bones. It's removed from the blood. You know what I'm saying? And when calcium levels go down, you release which hormone? PTH. Okay? So, homeostasis of bone. Oh my God. So, effects of aging, of course, you lose matrix and so forth. Bone loss consequences, you start getting fractures and whatnot, right? Okay, so muscle structure. Oh, we gotta move it on here. So this, of course, always starts with the function of that system. So we know what muscles do, movement, right? Because they're connected to the skeleton, right? And when they contract, they move the levers and then posture, of course, upright posture. There are certain muscles right now. If I'm standing upright, doesn't mean all my muscles are relaxed. They are my hamstrings, they are my quadriceps, my abdominal muscles, and my glutes keep me straight, like that. You can imagine if these muscles are contracting and these muscles are contracting, and I'm on it's a pole, so that pole just remains erect like this, straight up posture. So muscle, and then respiration. How is muscle involved in respiration? And remember what kind of muscle it is. Okay? So movement is of course going to be skeletal muscle, right? Because it's connected to the skeleton. And then posture is going to be also skeletal muscle. Okay? And then respiration. What's that? Respiration is going to be what kind of muscle? Smooth or skeletal? Not really. Because all those muscles for respiration are between the ribs. And what are ribs? Bones. Skeletal muscle. The diaphragm is attached to the rib cage. Skeletal muscle. Okay? So respiration is skeletal muscle. Okay? Because those intercostal muscles between your ribs are attached to the skeleton. Okay? So be able to explain each of these functions, right? You know, and talk about what kind of muscle is involved. Because we got cardiac, we got smooth, we got skeletal muscle. Okay? Heat, of course, is produced by skeletal muscles. Okay? And we talked about how that is. When energy from ATP is used up, not all of it is. Nothing is ever 100% efficient. So the excess heat, the excess energy is given off as heat. It's a byproduct of muscle contraction. And then the blood picks it up and then distributes it throughout the body. To maintain 37 degrees C. Communication, what am I doing right now? I'm talking. All the muscles, like all my jaws, skeletal muscle, all right, attached to my jaws, making my jaws move. My tongue, okay, it's also attached. The tongue is really a muscle, skeletal muscle, because it's connected to a muscle, a bone. The hyoid bone is underneath here. Okay, the hyoid bone is the one that doesn't articulate with any other bone. It just hangs and supports the tongue. Okay, so all that, and then of course the vocal cords. The respiration is what makes sound when air moves in and out of the trachea. The vocal cords are right there. When air is moving in and out, it's moving the vocal cords. So skeletal muscle again, because skeletal muscle is the one that's involved in increasing and decreasing the thoracic cavity volume, which sucks in air and pushes air out. So communication, you can say a lot of things about how the muscular system is involved. Okay? So, and then of course organ constriction, like the small intestine, stomach, and things like that, blood vessel, and the smooth muscle. Okay? Your body is about 40% skeletal muscle, so you can imagine all the functions are going to be skeletal muscle. Yeah? And then of course heartbeat is cardiac, and it's only found in the heart. All right? So that's an easy one too, in terms of the functions of the muscular system, functions of the skeletal system. Those are easy questions. You should be able to do those. And then these are the functional characteristics of muscle. It's got to be able to be excited, excitability. It's got to be able to be elastic, go back to its original shape. It's got to be able to be excited, excitable, so you can stimulate it. All right? And then, of course, contraction. It's got to be able to contract, contractibility. So those four are functional characteristics. They are characteristics of muscle that have a functional nature to them. They do something. And of course, we know about the skeletal, cardiac, and then of course they differ in structure, location, function, right? We just talked about those differences right now when we were describing the function of the muscular system, right? And then we know the smooth cells, they are the spindle shaped. Remember that table I showed you? I need you to know all those things. And then skeletal muscle fibers, those are long cells, right? Because they are long, they got a bunch of nuclei, they're multinucleated, the skeletal muscle cells or muscle fibers. If you call them muscle fibers, it's easier because then you know what the shape of it is. Okay? If you call them skeletal muscle fibers. Right? And then activation, of course, they differ in activation. Smooth muscle can respond to hormones and things like that. But skeletal muscle doesn't, and it's involuntary. It's voluntarily controlled for the most part. Skeletal muscle. Smooth muscle is automatically controlled by the autonomic system, so it's involuntary for the most part, right? And it also, and the heart muscle takes it to an even new level. Cardiac muscle is self-exciting, it excites itself. It's autorhythmic, things like that. Okay, so for these differences between the smooth skeletal and cardiac muscle, in terms of the cell shape, the number of nuclei, and where the nuclei is located and things like that. It's all in that little table that I showed you. Okay, so sometimes I ask about those differences, right? And then, of course, muscle is composed of muscle tissue. It's got nervous tissue, connective tissue, right? Because muscle, after all, is these proteins that make up muscle bundled up in connective tissue membranes. That's it. That's what muscle is. And then, of course, you need the nervous tissue to excite it, to stimulate it. So I know about that. And then, again, the connective tissue that we collectively call fascia. Okay. Now, mice, sarco, all those are the prefixes for muscle. We just finished osteo and chondro for the bone and cartilage, right? So epimysium would be on the outside, covering the whole muscle, and then the perimysium will be the one that covers the sub-bundles, called fascicles. They're covered by the perimysium. And of course, along all these connective tissue runs blood vessels, bringing in the blood and nutrients and so forth, and gas, oxygen. And then endomysium, okay, covers the muscle fiber, right? Remember we talked about how the muscle fiber, the muscle cell, has just the plasma membrane with those phospholipids, and it's not strong enough for something that's going to be pulled apart and contracting and doing all sorts of stuff. So it needs a connective tissue layer, so it kind of has an outside jacket on the cell itself. Then the cells are bundled up and form fascicles, and then they're covered by perimycium. And then the fascicles come together to form a whole muscle, and it's covered by the epimycium on the outside. Right? So that's all muscle is. All these compartmentalizations by the connective tissue membranes. So gotta know those. And then of course the terminology, like I just told you, right now we got sarco meaning flesh. So anything to do with the muscle cell, like the cytoplasm of a muscle cell, is called the sarcoplasm. The endoplasmic reticulum of muscle cells, sarcoplasmic reticulum, right? And then another, mice, like the epimycium and so forth. You know what I'm saying? What's another prefix? Myo, like myosin, which is the thick protein in the muscle. Okay? Right, so know that terminology, and then of course actin and myosin, those are the myofilaments. Actin is the thin or thick filaments. Actin?
Oh, thin.
Thin. Okay, and then myosin is okay. And we know how they are arranged. We got the Z line here, we got the Z line there. Okay? And then the thick filaments are in the middle like that. Okay? And those are what forms the bands and the striations and so forth. Okay? Okay, so we get the sarcomere, which is that from one Z line to one Z line, and it's got actin and myosin filaments together. Okay? And then it forms the I band. Okay? Okay? I'll draw it again. So when a muscle is relaxed, they only overlap slightly. The actin and the myosin, okay? They overlap slightly, okay? And then this is, what protein is this that I'm drawing? What protein is the blue one? Okay. The blue one is called titin, okay? It's an elastic protein that just sort of anchors my myosin right there. Okay. Okay. And the red one is actin. Those are the thin filaments, right? Okay. And right here at the Z line, at the Z disc, what protein do we have? Connectins, right? Okay, those kind of disc-shaped proteins. And then what protein? And then here, we got the M line. Okay? And then we got jasmine. Kind of anchors that in place, the myosin filaments. So that when the muscle is contracting, what's happening? The Z lines are approaching the center, right? They're moving towards the myosin. So from here to there is the I band, right? And from there to here is the A band, okay? And from here to there is the H zone, right? Okay. So when we are contracting, what's becoming smaller?
IH.
The I is getting smaller here. The H zone is getting smaller here. In fact, the H zone will disappear when these overlap the actin. And then the sarcomere is also getting smaller because the Z line is moving towards the myosin, right? so that when the muscle is fully contracted, it's almost the length of the A band, which is -A, right? Okay, so I want you to know those proteins, right? And you know the structure, which is going to be important for muscle contraction. You know, when you talk about muscle contraction, because when that happens, this is moving inwards, and the actin is sliding past the myosin, right? And they're not changing length. The actin is going to remain the same length. The myosin is going to remain the same length. Okay. All right. So here I want you to know the bands and the bands that get small. I think that's okay. You get a question? You can stop me because I have to rest a little bit here, asking questions. Okay? I'm kind of going fast because, you know, we're just reviewing. I'm trying to point you to the things you need to know. So that would be a good kind of summary to go on when you're studying for those, you know what I'm saying? For your exam. And the things that I tell you are coming are definitely coming. You know what I'm saying? So if I were you, sometimes I would even go to that integrity and listen again and study those things, because I will bring them. I don't, you know, just blow hot air. No, I don't do that. So sliding filaments that I was talking about, the actin and myosin same length, they're just sliding past each other. All right, and I bend, axon, and sarcomere gets short. The change in length. All right. And then, of course, the A band remains the same. Okay? It's constant. I need you to know which bands. In fact, the arrangement of the actin and myosin is what gives the striations. All right? So, relaxed state, thick, thin filaments overlap slightly. When it's contracted, they overlap even more. Right? And then, muscle physiology. All right? Muscle physiology talks about how an action potential or a nerve impulse will stimulate the muscle to contract. So it involves events at the neuromuscular junction. This is actually a short one. So we talked about how the membrane of the muscle, the membrane of the axon fiber, they are polarized, which means the charges are separated so that the inside is more negative, the outside is more positive. Okay? Based on the number of ions on the outside versus the inside. So you have more positive ions on the outside, okay? And the inside is negative because some of the other big negative proteins do not exit the cell because there are no channels for it. So it gives the inside a negative charge. All right? So that's resting potential, which is minus 70 millivolts, right? And then we have these channels or these membranes that allow ions to move so that we can depolarize. We can undo the polarization. We switch it so that outside is more negative, inside is more positive, and that's depolarization, right? So a ligand-gated channel will open when something attaches to it, like a ligand, like acetylcholine, right? So know those two kinds of gates. One that opens when something attaches to it, which is a ligand, like acetylcholine. One that opens when a voltage changes, so it's voltage gated. So it will listen to, say, minus 55, for instance. Those sodium voltage gated channels, when you reach threshold, a whole bunch of them open up, and then you depolarize. Okay? And then the potassium channels listen to plus 30. They open up at plus 30, and also the sodium ones close at plus 30. Okay? So all those channels are voltage gated because they listen to the voltage. They respond to changes in voltage. Okay? So be able to explain those two kinds of channels. I might say, okay, what are the two kinds of ion channels that we find in the muscles, in nerves? You talk about ligand gated, and you talk about voltage gated. So, okay, that way we do that. And then, of course, action potential is always okay when you get to minus 55 millivolts. And we get there because these open ligand-gated channels. When the impulse comes in, acetylcholine is released, and we get the ligand-gated channels are the ones that get us to threshold. Then, after that, then the voltage-gated sodium channels open up because they listen to a voltage. All these ligand-gated channels do is take us to minus 55. At that point, voltage-gated channels will take over. Okay? If you don't get to threshold, what happens? nothing. It's an all or none action, okay? So that's the action, but it's all or none. And then, of course, depolarization occurs if you reach 55, which is brought about by ligand-gated channels. And then the voltage-gated channels take over after that, and they take us through depolarization to plus 30, and then repolarization back down to rest, okay? By repolarization, when potassium ions move out, depolarization, and sodium channels move in, sodium ions move in, okay? All right, so gotta know that this is important in terms of, so nerve stimulation is done by motor neurons, okay? They cause the muscle to do something, to contract, so it's a motor activity. Motor neurons do that. And then, of course, these motor neurons, we talked about how they are these kind of cells, right? And then nucleus. And then the fiber of it, which is the nerve fiber, is really the axon, which is a one long process coming from the cell body. These shorter processes are called dendrites. So this is what we're saying travels in nerves. So what is a nerve? It's a bundle of axons. That's all a nerve is. You know, when we do the muscle dissection, you'll see a bunch of nerves too. The other day I was showing the other guys in AMG2 nerves. And they're big. You can't even miss, even in a cat. Okay? And because there's a bundle of all these nerves, okay, and covered by kind of connective tissue, and sometimes they're covered by myelin sheath. But right now, just know that the axons will travel in the nerves. Okay, so axons, we talked about how a muscle is a whole bunch of cells, so it's going to branch profusely, like a lot. You can get tens of thousands of branches on one nerve fiber, okay? On one axon. You can control 2,000, 10,000, muscle cells. So they branch profusely so that you don't need a lot of this wiring, if you will, really. So the events at the neuromuscular junction, right? The neuromuscular junction, we talked about it. We have the synaptic knob like that, and then you have a special part of the membrane with the receptors, right? With the ligands, right? Sodium ligand gated channels that will respond to acetylcholine, right? Okay, so we talked about how you have synaptic vesicles in here. What are they containing? It's the synaptic terminal. Yeah, acetylcholine, all right? And then, so know what these are. And then, of course, the motor end plate, we said, is the special membrane, or the sarcolemma. Remember, the sarcolemma is the muscle fiber membrane that has receptors, the ligand-gated channels, and also acetylcholinesterase, right? Acetylcholinesterase is also waiting here because it's going to do what? Break down the acetylcholine when we're all done. Okay, this doesn't touch. Okay, so that's what the motor end plate is. It's a special part of the sarcolemma or muscle fiber membrane that has receptors, ligand-gated, sodium ligand-gated channels, and acetylcholinesterase that breaks down acetylcholine. And then the cleft we said is the, what is the cleft? The physical gap between the two, because they don't touch. Because they don't touch, they're not physically connected. So what is the connection between the two? It's functional. Because the nerve can affect what happens here by sending a messenger called acetylcholine. So it's that functional connection is what we call synapse. Okay? So know those terms, okay? Be able to describe those. Sometimes I might put them in the short answer and say, describe what is a synapse, what, at the neuromuscular junction, what is, or I just say synapse, you should know that it's a neuromuscular junction. What is the motor end plate? Okay? The sarcolemma with the receptors, okay? And then the events of the neuromuscular junction, which was most of the lecture, right? When you get the impulse coming in, calcium gets in there, attaches to the vesicles, exocytosis occurs, acetylcholine sprinkled in the synaptic cleft, right? And then where does it go? To the ligand-gated sodium channels. And then they take us to threshold. They open up, sodium goes in. And all these events are occurring during the leg phase, right? Okay? So all those events, and then once the threshold is reached, they close up, acetylcholine is removed, and then acetylcholine is recycled at the neuromuscular junction, right? Because we've got acetylcholine that's going to break it down, right? To the choline, and then the choline is transported back into the synaptic knob by a symport, a sodium symport, right? And then it is symported, choline is symported in. What happens to the acetic acid? It just diffuses in the blood. Because the cell in the synaptic knob are going to be producing acetic acid anyway, because of the cell metabolism. So you don't need to transport it back in there. Okay? It is just metabolized in the liver, okay? And you then reassemble acetic acid and choline from acetylcholine, and then you package it into the vesicles and they wait for the next impulse. So all those 9 events, you could get a bumper harvest of points on that. So there's all those events, you'll be able to talk about 9 of them. So this is the last lecture, that was the last one. So what is excitation contraction coupling? You are coupling the excitation when the impulse comes in, and then the muscle does something, it contracts. So you are coupling the excitation and the contraction, and together, because it's the excitation that causes the contraction, because the muscle is excitable. Remember those functional characteristics, and this is contractibility, which can contract. So this is stimulation, and then of course propagation along the nerve. That's how the nerve impulse travels, the action potentials. All right. And then what's the deal here? What we want to do is, again, calcium is very important here. Calcium is in bone, and calcium is used in this. And then the calcium is stored in the sarcoplasmic reticulum. Remember the functions of the sarcoplasmic reticulum in muscle cells is to store calcium. Okay, so when the impulse comes, it will open those calcium channels so that calcium goes into the cytoplasm. Okay, because calcium is going to be attaching to troponin. Remember that muscle that hangs us tropomyosin on actin? Remember when you did muscle structure, right? Okay, so that's the whole purpose of bringing in the impulse. All right. So that process will result in contraction. So it's excitation, contraction, coupling. Okay. All right. So we've got to distribute the impulse throughout the bundles of muscle cells, muscle fibers, right? So how do we do that? If we only have the membrane on the outside only, okay, so say this is a bundle. Okay, these are the muscle bundles, right? Muscle cells. And this is the outside membrane. If you only stimulate the outside, are the bundles on the inside going to be stimulated? No. So what happens? What happens is that the muscle fiber membrane, the sarcolemma, will invaginate inside, make its way into the deeper layers as T-tubules. Okay? You see what I'm saying? Those two, the T-tubules are actually invaginations of the sarcolemma, so that the impulse is taken to the inside, so that inside bundles will get it. And it so happens that along that, the T-tubule, on either side of which will be the sustaining of the sarcoplasmic reticulum. Because what's in there is the calcium, because we need the calcium to open up channels, and then the sarcoplasmic reticulum will release calcium to the cytoplasm, right? So that it will go and attach to those actins, at the troponin on the actin, right? So that whole assembly, where you have the cysteine on the outside here, and then the membrane on the outside here, this is the membrane, right? The sarcolemma. So, and that's the triad. Because it's got two cysteine with the calcium inside here, calcium. Okay? And then this is carrying the impulse so that it excites all the calcium channels along the sarcoplasmic reticulum. So sometimes I say what describes the triad, we talk about the two cysteine, which are, extensions of the sarcoplasmic reticulum carrying calcium. Right? And then the extensions of the membrane, the muscle fiber membrane, are the T-tubules. And they make the three structures triad. Okay? So that's how you stimulate the muscle to contract. So the impulse is spread through by the triad, because the T-tubules, the membrane will go inside. And then, of course, it will cause the depolarization and then of the membrane, and then the calcium gates open up. Okay? Just like the calcium gates opened up at the neuromuscular junction in the synaptic knob. And then the calcium will attach to troponin. Remember we said troponin anchors a tropomyosin, which is a fibrous protein. covers the binding sites. When calcium attaches to troponin, troponin changes shape, and then it moves tropomyosin out of the way so that you expose the binding sites. The active sites on G-actin, remember G-actin is a globular actin, okay? So it exposes the active sites on G-actin, right? And then once the active sites are exposed, those cross bridges on the myosin will attach, okay? I think there's no class today coming. Okay, so when you want the muscle to relax, you have to get rid of the calcium, right? And that takes energy to sweep off all the calcium so that troponin goes back to its original shape. When it does that, it will move tropomyosin and then so that it's now covering the binding sites. As long as the binding sites are covered, the muscle will not contract because the myosin heads will not attach. You can't form cross bridges. And then the troponin will reverse to its original shape, and then the muscle will passively just relax. Remember, it's elastic. Will just passively relax, okay? And go back to the original length. All right? And then, so a muscle twitch is the contractile response of a muscle, you know, in terms of few muscle fibers contracting or a whole bunch of them, okay? And it's got leg phase when you sort of set and you produce, the calcium is released and one node, and then the contraction phase starts where the cross bridges form, and then the relaxation phase starts when you start to pump calcium out. So those are the three phases of the muscle twitch, right? Okay. A motor unit is simply the neuron, okay, and the muscle fibers that it controls. Remember, one neuron will branch profusely so it can control up to 2,000, 10,000 muscle cells. So that unit of the muscle fibers that are being controlled in that one motor neuron, that's what is a motor unit. Okay? When you are lifting heavier and heavier weights, you start to stimulate more motor units so that more muscle cells will contract to lift the heavier weight. Right? So when you reach your heaviest weight, you have stimulated all your motor units and you have none more to do more work. Okay? So motor units are recruited when you do more work, when the stimulus is getting stronger, all right? And then multiple motor unit summation is when you have more motor units being called up to do more work, all right? And then, so stimuli of increasing frequency, let's say you get a stimuli comes and then another one comes close behind it, okay? The muscle, when it contracts with greater force, okay? So that's multiple wave summation, okay? So that's higher frequency of stimulation. You're stimulating at high frequency, and then it will contract with greater force because the muscle is stimulated many times per unit time. Okay. Oh my God. Is somebody coming? And here you're getting increased stimulus strength. It's really going high, and then the muscle will simply contract with more force. Okay, because it will recall more motor units. Okay, you see the difference between increasing strength of stimuli and frequency. Frequency will just cause the muscle to contract with greater force. And the same motor units are contracting, but just with greater force. But here, when the stimulus is very, very, very strong, it reaches way more than threshold, then you have more motor units coming in to do the job. Okay? That's what it is. So note the difference between multiple motor units, and because the name tells you anyway, and then multiple wave summation, okay? Multiple wave, it means you're getting many frequency, you're stimulating the muscle many times per unit time. And then incomplete tetanus is when the muscle contracts without fully relaxing, okay? So it's going to contract again, so that's incomplete tetanus. And then complete tetanus, the muscle stays contracted. It doesn't relax, there's no relaxation. And then it becomes painful at the end. That's when you get a cramp. Because the calcium is not being swept away, okay guys? I know this is a lot of information, But and then trap is, what happens in trap is that when you apply a stimulus of maximum strength, and then you let the muscle relax, and then you apply another one, the muscle will successively contract with greater force. Okay. Then on the third or fourth one, it will contract with the maximum force all the time. Okay. So you trap, you're applying maximum stimulation, and you're letting the muscle contract and relax. Then you apply another one, then another one. So if you do that, the muscle will have like a grade like this, and then it contracts, and then it will start to contract at maximum like that throughout, because you're letting it contract and relax. So be able to explain incomplete tetanus, complete tetanus, and trap or trapping. Okay. So these are just different kinds of muscle contraction. So isometric, metric is length. So if the muscle contracts at the same length but different tension. Okay? Isotonic, tonic is tension. So iso means same, so it's same tension but changes in length. Okay? So an eccentric contraction is in the isotonic contraction where the muscle, it's got the same tension but it's lengthening. Like when you're doing bicep curl and you're dropping the weight, your muscle is lengthening, but because you're carrying the same 5-pound weight, the tension doesn't change. And then a concentric is an isotonic contraction where you have the same tension, but you're doing your bicep curl going up, your muscle is shortening, right? But the same tension because you still got the same 5-pound weight. Okay? So just to be able to describe these kinds of contractions. And then muscle tone is a state of partial contraction of the muscles. Okay? State of partial contraction of the muscles. Okay? So that's why when you work out, your muscles become tight, because that's when you say, I want to get toned by going to the gym. Because they end up being slightly contracted, which makes them more firm. Okay? So that's what muscle tone is. And then, of course, asynchronous contractions is certain motor units contract while others are resting. So that allows you to do more work. Okay? But if you overstimulate and all the motor units are being called up, then you get tired, you get to fatigue, which is what we're doing now. Fatigue is the inability to contract of a muscle, and it is CNS psychological. If you perceive in your mind that I can't do this work, and then that's psychological fatigue, and it starts at the CNS. So if somebody's running a marathon and then at the end of the race, somebody's trying to come back and beat them, and then you give your last burst of energy, you are mentally beating psychological fatigue, because you are thinking you're tired, but you are putting a burst of energy. And then ATP depletion causes muscular fatigue, because the muscle simply doesn't have the energy to remove calcium and so forth, and the muscle relaxes, and then you start to get cramp, which is like physiological contracture. Your muscle doesn't have any more energy. Remember to sweep the calcium back into the sarcoplasmic reticulum. It's an active process requiring energy. So when you're tired, you don't have any more ATP, so you cannot sweep the calcium back into the sarcoplasmic reticulum. And as long as the calcium is there, it's going to be attached to troponin, and the muscle is going to stay contracted. So that's when you start to get cramps at the end of your exercise, because there's no longer enough ATP to move the calcium out of the way so that your muscle can relax. You get the Charlie horse, you get the cramp. Okay? And that's physiological contracture. And this happens, rigor mortis is like physiological contracture that happens when you die in the first few hours, because there's no longer the energy, you're not producing energy, so the muscle remains contracted like that. And then after a while, the muscle denatures, and then you come in, and then, that's it. Right? Okay, so just know those two processes. And then this one just basically shows the sources of energy, where creatine stores energy for a little bit, for like 10 seconds, and it gives 1 ATP for 1 creatine. And then anaerobic is glycolysis, which is breakdown of glucose to pyruvate, and it gives 2 ATP. It's about 3 or so minutes. And then the one that gives the most is 34 ATP for aerobic respiration, where you use oxygen, you go to the mitochondria via oxidative phosphorylation, okay, in the Krebs cycle, and it uses amino acids, fats, glucose, everything to form the energy. So you form 30, and it lasts for hours. So this will be in a long, in a endurance athlete, they have more aerobic muscle fibers, and then a power athlete will have more of those glycolytic. And because glycolytic is inefficient in providing ATP, only 2, but it's fast. Okay, so now we couple the energy source and the speed of contraction. So slow twitch, oxidative, because oxidative, while it makes 34 ATP, it's a process. But then we go through all that Krebs cycle and form, it's a slow process. So they are slow twitching muscles. So there will be slow twitching and they're more endurance. So SO is more endurance, and then fast twitch glycolytic. Remember I said glycolysis breaks down glucose, forms pyruvate and then lactic acid, and you only get 2 ATP. It's inefficient because it produces 2 ATP, but it's fast. So those are fast twitch, okay? And so sprinters like, Bolt, they have a lot of these glycolytic fibers, which are big and fat, because they're not fat, but they are big diameter, because they've got glycogen storage in them, all right? And the slow twitch are thin and narrow because they don't have to store any glycogen in it, and they have blood vessels. They are the red muscles, so these are red. And these are the white muscle fibers, the glycolytic ones, because they don't have a lot of blood vessels and so forth. And then fast twitch, oxidative, they are kind of in between, 4. They use both oxidative and anaerobic energy, glycolytic energy. Okay? So you can kind of go, this is just showing hypertrophy growth in size of muscles, and by increasing the myofilaments inside, the bundles inside. And then atrophy is decrease in size. Have you seen people in a wheelchair that are not using their muscles? After a while, their muscles get smaller because they're not being used. Whenever you're not using your muscles, that's why if you work out, your muscles get bigger because they're being used. They develop more fibers in them. Okay? So, by the time you're 80, you've lost 50% of your muscle mass. And then everything kind of slows down. And your fast twitch fibers go quickly too. You can respond slowly all the time. Okay? Then smooth muscle, there is visceral smooth muscle, which is mostly the small intestine and the stomach and whatnot. And multi-unit kind of contracts as different units, as groups of fibers. Like the iris of your eyes, smooth, some blood vessels, you have bundles of these multi-unit smooth muscles. Okay. And then regulation, multi-unit nerves, hormones, etc. Visceral is usually autorhythmic. Oh my God, you gotta go. All right, and then this is structure of muscle cells, a smooth muscle, spindle-shaped fibers, and then functional properties, and that's it. Sorry guys, we had to take a little longer, but okay, I'm gonna have to give you a few minutes to get to, I'll give you 15 minutes and we'll start the lab. Anyway, today I'm just gonna do a quick introduction to the electrophysiology equipment. We don't have much to go, okay?